Hi, good afternoon, everybody. All right, I'm actually going to start with a couple of disclaimers. One is, I know this conference is about AI, but I'm not really going to be talking about AI and personalized medicine.
Hold the microphone closer.
Is this better?
Much better.
Thank you. So I'm going to be talking about instead using large patient data sets to really accelerate scientific discovery in the field of precision medicine. And so in some sense, it's pre-AI. So I do believe that AI will eventually be able to help in this area as well. And the second thing is, I think one of the panelists in the previous panel talked about autonomous cars not being an industry and being a science project. Well, you're about to see a science project now. So, you know, hopefully you'll still find it interesting. So let's get started. So a very high level, let's start at the basics, right? So what is precision medicine? Precision medicine is really the ability to tailor treatments or even kind of determine prevention strategies based on the genetic characteristics or more generally the individual characteristics of a patient. And it can take on many forms. People have been talking about it, especially in the last few years, about how to do it. But I'm going to focus more on the genetic angle of it. So some of the forms it can take is the ability to figure out if certain patients can tolerate certain medicines. So for example, warfarin, which is a blood thinning agent, is often used in the treatment of heart disease, and it works really well, except if you have certain mutations, in which case you need to really lower the dose or you could actually die from excessive bleeding. So that's where you realize that genetics is very important in deciding how you would treat a patient. You could also have genetic tests to determine if you have a higher kind of probability of developing cancer. So probably the most high-profile case would be Angelina Jolie, who underwent preventive surgery because she had a mutation in a gene called BRCA1 that put her in a higher risk for developing ovarian cancer and breast cancer. And the last one is actually is something that I'm very focused on, and that is to actually use the genetics of a patient's cancer to figure out new treatments for the patient. And so probably the biggest shout out for precision medicine came from President Obama in his 2015 State of the Union address, where he announced the Precision Medicine Initiative that allocated about $215 million for this effort. And I'm one of the beneficiaries of that in the form of federal grants. But the amazing thing is, in this world where there hasn't been a lot of bipartisan support on many initiatives, this is one of the few that has a lot of support from both parties, basically. So let's step back a little bit and now look at precision oncology, which is basically precision medicine applied to cancer. But we all know about cancer. We have all been kind of affected by people who have we have like friends and neighbors who have had cancer. But what does it really mean at the cellular level or at the genetic level? So cancer is essentially a case of uncontrolled cell division. So what happens is that in normal cells, you have checks and balances that make sure that eventually cell division stops. Whereas in the case of cancer, those checks and balances disappear because of problems in the DNA. And what ends up happening is that the cells keep growing, and you have this new tumor mass or neoplasia or cancer, whatever name you want to call it. So what does it mean at the genetic level, right? So what it means is that there are really widespread changes in the DNA and the RNA. Again, I'm throwing out acronyms here. I'm going to define some of them as I go along. And that's what cancer is. And the goal of precision oncology is to really try and understand how to use these genetic changes to tailor treatments for patients. So stepping back, I don't know how many of you have looked at biology after high school. I know I didn't until I started working in this field about 5 years ago. So the central dogma of biology is this three-step process where you have DNA. DNA is essentially your genetic code. The DNA gets converted into something called messenger RNA or mRNA through a process of transcription, and then finally you end up with proteins. From messenger RNA to proteins via a process of translation. And proteins are the workhorses in your cell, and they are basically doing all the work. Now, what happens in the case of cancer is you have disruptions at each of these 3 levels. And so one of our goals is to try and really create a catalog of all of these changes. And so what do the changes look like, right? So at the DNA level, you have something called somatic mutations. Now, somatic mutations are alterations in the DNA that happen after conception. So these are things you don't inherit from your parents. This is happening all the time. It's part of our aging process. Most of them do not result in anything harmful, but if you're one of the unlucky few, some of these mutations could actually end up being cancerous and result in the initiation and possibly progression of cancer. And so the way to think of a mutation is really spelling mistakes, right? I mean, some spelling mistakes don't matter, but in some cases, you make a spelling mistake in public, remove the L, and it means something completely different, right? And that's what ends up with mutations in cancer also. Most of them don't harm you. So that's one. Then the other one is something that's known as copy number alterations. So all of you, you have 23 pairs of chromosomes, and so each pair has a copy of a gene. Now, most of the time, that's fine. That's what most of us have. But every once in a while, for some reason, you either end up losing some genes by a deletion even, which is kind of the figure on the left, or you end up kind of having more copies of a gene by process of duplication. Again, you see lots of these in cancer. The challenge is to figure out which ones of these are important. So these are both changes at the DNA level. If you go back to the central dogma of biology, that's the first step. You also see changes in the RNA itself, which is the messenger RNA step. And again, if you look at a normal cell versus a cancer cell, you see 2 sorts of changes. Either you see a lot of overexpression of the mRNA, which would actually result in more protein being formed, or you would see underexpression, like loss of the mRNA, in which case you wouldn't have any protein. And in many cases, there are enough compensatory circuits and redundant pathways where if one particular protein is not being produced, it's fine. But once in a while, again, you get unlucky, and this could end up being a critical protein, and that could really affect you and result in the development of cancer. And there's something else called epigenetic changes. I don't really want to get into that, but this is an entirely new category of changes that can again cause problems in mRNA formation. So why am I talking to you about all of these changes, right? The idea is, we want to really be able to catalog all of these changes, because the first step to really understanding the problem or trying to solve the problem is to actually list out all the changes that we have. And this is something that, cancer biologists knew about this. Like, yes, there are each of these changes, but what ended up happening is because of the falling costs of DNA sequencing, a lot of cancer genome projects started. The one that I'm showing here is something called the Cancer Genome Atlas, which was funded by the NIH. And the idea there was to just really create a detailed molecular map of all the changes genome-wide. So we have 20,000 genes. So for each of the 20,000 genes, they take a cancer patient and they measure the different changes that are there. And so at this point, this project has about 33 different tumor types. It has around 11,000 tumors and a ton of data. Again, it's nothing compared to what Paul just talked about in the previous talk, but given the difficulties in measuring each of these changes, it's amazing that we have this amount of data. And this is just the beginning. There are new cancer sequencing, genome sequencing consortiums being formed. So there's something called the ICGC, which is really the international consortium that looks at data from cancer patients across the world, and they're trying to do something similar. So why am I so excited about this data? Because I'm a computer scientist, and before this data was available, I would not be able to work in this field, because I would have to rely on whatever data is given to me by biologists. But right now, I can just go to this database, which is publicly available, download it, and apply my own cool machine learning technique and try to solve some problems here. And so that's essentially, in some sense, what we did. I started this work. The work I'm going to present to you started about 5 years ago. And hopefully, it's still a work in progress, but hopefully it'll be exciting for you guys. So I'm going to skip over this, but this is something that's available that shows what was the utility of kind of doing all of this data collection. So one of the big things that came out was the realization that cancer should not be classified by the tissue of origin. So normally now, if you have lung cancer, you go to a certain kind of oncologist. If you have breast cancer, you go to a completely different kind of oncologist. But what they realized is tumors are actually very similar based on their genetic characteristics. So they actually realized that a certain type of breast cancer is actually very similar to ovarian cancer if you look at the underlying genetic changes. And they also realized the opposite thing, which is just because you have lung cancer doesn't mean you're identical to another lung cancer patient. So what they found out was there were different subtypes in cancer. And so the important thing was to go after the genetic characteristics of the tumor rather than focusing on the tissue of origin, even though that is somewhat important, but it's not the key criteria to focus on. So this is, as I said, 2005 was when all of this data became available. People are still churning it, making sense of it. I know 10 years in the tech space feels like a lifetime. 10 years in sciences is not that long. So things are moving, but surely enough, they're moving in the right direction, I think. So my focus, again, there could be many different ways you can slice and dice this data. So my focus has really been to focus on the precision oncology aspect of it and try to mine the large data sets to generate hypotheses. And once I'm somewhat satisfied about the hypotheses I've generated based on existing literature, I go and I kind of track down cancer biologists who would be willing to validate my hypotheses. And so far, this has worked pretty well for me. And so I'm looking forward to kind of continuing along that path. So this, again, kind of going into the details a little bit more, the specific problem I work on is known as mutation-specific therapy. So the idea is you look for specific mutations in the cancer, and then you try to identify drugs that would actually target the genes underlying those mutations. This is a big difference from what happens in the clinic today with chemotherapy, where in chemotherapy, you basically try to target all rapidly dividing cells. If you remember in the beginning, I told you that cancer is a case of uncontrolled cell division. So what chemotherapy does is it goes after everything that is rapidly dividing, which includes your hair cells and like cells in the stomach, which is why you end up with some of the toxic effects of chemotherapy. But with this mutation-specific therapy, because it's very targeted and it's only going after the cells that have the mutations, the hope is, and the evidence so far is that it's going to be much less toxic. But the issue and the challenge that is there is to really identify many more of these mutation-specific therapies, right? And so that's where we sort of come in. And what we decided was to actually look at a technique of like compensatory pathways. And so this, for those of you, with engineering backgrounds, this is basically redundancy. If you have an electric circuit and you have two parallel paths, if one of the paths gets conked out for some reason, you would still have current flowing through the other path. But if both of them go, then you have an open circuit, right? And so that's what happens with this concept of compensatory pathways in biology. The name of this phenomenon is synthetic lethality. So if you look at this cartoon over here, imagine you have a pair of genes A and B. Now, when A goes down, B is able to keep the cell alive. When B goes down, A is able to keep the cell alive. But if both of them go down, then you have a dead cell. Then you have a dead cell, basically. And so, in the case of cancer, imagine that one of the cells is one of the genes is actually the cancer mutation. Its functionality has been knocked down by the mutation. Now, if you can find its synthetic lethal partner, gene A, you would be able to actually selectively kill only the cancer cells and not the normal cells. So I'm being told I'm a little short on time, so I'm going to skip over some of the details of the algorithm, but I want to point out that the key contribution that this work has done is to kind of develop a method for kind of expediting the large-scale identification of synthetic lethal partners using cancer genomes, and this is something that was not done in the past. In the past, people would rely on experimental approaches, but this is the first kind of purely data mining-based approach that tries to find these synthetic lethal partners. I'm going to skip over this. So what we have is a tool, we've called it Methyl. It's a play on an actual Methyl because we hope that it's going to be as precise as real Methyls, and in basically targeting just the mutated cancer cells. And just to kind of give you what the input and output is, so you start with a mutation and a cancer of interest. So the cancer of interest could be leukemia or breast cancer or whatever. And then you run it through this software, and you basically come up with a short list of synthetic lethal candidates. And then the idea is you would try to find drugs that would target these synthetic lethal candidates or genes, and that's the way you come up with drugs that are specific to these mutations. The details of how we showed that this method works is actually in a paper that we published in Nature Communications. I'm going to skip over that. But the most exciting thing was we were actually able to use this tool to make a new discovery. It's one thing to do both either retrospective or prospective validation, but we were actually able to find a new synthetic lethal interaction that was not known previously. So what this means is that patients who have the IDH1 have this particular gene IDH1 mutated in leukemia could potentially have a therapy. We are still in the process of validating it because obviously when you have to get to therapy, that takes many more steps of validation. But we were able to validate it using patient cells and implanting them in mice. So that's still. It's more advanced than most of the tools have gotten. So this is just showing that, this is not just specific to this IDH1 mutation in leukemia. We are able to find therapeutic options for many mutations in different cancers. So where am I headed with this, right? So one is my goal is to get this in the hands of cancer biologists. And right now, we have a lot of ongoing collaborations with oncologists at MD Anderson, Stanford Medicine, and hopefully, and as you can see, some of these cancers are pretty aggressive and hard to treat. So hopefully, some of this will actually kind of pan out and give rise to new treatments. But most importantly, actually, as a result of these experiments, we are hoping to be able to get enough of the ground truth where we can start using machine learning techniques and AI techniques to be able to improve the prediction accuracy of Methyl. And in the process, reduce the need for experimental validation or reduce the number of candidates that we have to experimentally validate. There's another aspect of precision oncology where you try to identify responders for a new cancer drug. I'm going to skip over that because we are a little short on time. But again, we could essentially use Methyl in reverse to be able to identify the responders for these drugs. And I have a project going on at SRI International. We just got some federal funding where we would use Methyl to identify responders for a new class of drugs that have been developed at SRI International. And again, it's not specific to the drug. We should be able to generalize it to other kinds of drugs as well. And finally, I want to end with this vision of what a precision oncology flow would look like. The idea would be you would have a cancer patient, comes into the clinic, you get the tumor DNA sequenced, and you find out the unique mutations that the patient has, like the somatic mutations that the cancer has actually. And then you harness Methyl to predict effective drugs. Again, these are computation predictions. You can't obviously give the drugs to the patient without some testing, but you should be able to test them in what's known as patient avatars. And then finally, from your computation predictions, you have a small set of drugs that now you've actually validated in patient avatars, and that you can give to the patient, and hopefully, that helps in curing his cancer. And if because of his or her bad luck, the cancer mutates and becomes slightly different, which often happens, you again repeat the process. You again sequence the DNA of the new tumors, and then you repeat the process. And what is so exciting about this flow is that as more and more data comes in, the Methyl is going to get more and more accurate, and so there's a positive feedback loop of continual learning that's going to happen. So I'm like really out of time at this point, but this is the vision I have, and this is what I'm working towards. And I'd be happy to chat with you if you have any questions along this. So thank you very much for your attention.